and my project is GEMINI, a breakthrough system for robust genetic interaction discovery, revolutionizing biological insight for the discovery of novel gene regulatory networks and the application of them to industrial-scale genetic engineering. So, genetic engineering is a very interesting field of science that essentially tells us that we can use engineering principles to design and construct new biological systems. This allows us to manipulate organisms or the genetic material of them to create novel functions that are not found in nature, with applications in medicine, agriculture, and manufacturing. Think of things you see in your everyday life, like insulin to treat type 2 diabetes. Those are all a product of genetic engineering. So, gene regulatory networks refer to this idea or notion that there are connections between genes or relationships between genes. For example, if gene A had an influence on gene B, and if gene B had an influence on gene C, manipulating gene B would inadvertently be disrupting the effect that gene A had on gene C, and those connections can all be modeled by a GRN or gene regulatory network. Now, the vast majority of genetically engineered strains actually fail in the industrial setting due to disruptions in this genetic regulation. Disregarded interactions between genes often account for various unintended effects, like ineffective metabolic engineering, etc. This limits the practical applicability of modification and manipulation techniques. Now, research finds that GRN manipulation significantly boosts performance efficiency and yield. The problem is that there's currently no fast and efficient way to infer expression-based GRNs in biologically relevant organisms. Current algorithms would take over 2 continuous years to infer a full GRN of the human genome, that too at a low accuracy. There are currently no full GRNs for any organisms because of this, a significant bottleneck in the biomanufacturing industry. So there are a couple different reasons for this problem, and the first is because the complexity of calculation explodes exponentially with each gene added. So if we have a 2,000 gene genome, we're looking at 2,000 squared interactions, and the complexity explodes exponentially. Additionally, traditional algorithms rely extensively on expensive supercomputers, and computation would take 2 continuous years with current algorithms on a standard CPU. There's no accurate way to construct GRNs because current algorithms cannot account for the complexity and nonlinearity of interactions, and current methods would result in an overprediction of null interactions. So my goal was to develop an end-to-end novel GRN inference system to achieve high speed, accuracy, and robust GRN construction. The criteria were accuracy, efficiency, robustness, practicality, and cost, and the constraints were both technical, functional, and resource constraints, with memory, parallelization, feasibility, and data availability constraints. So my project was split into 4 major parts. The first was gene expression measurement. The second was the construction of a parallel mutual information algorithm. The third was the construction of a GNN to predict network structure. And fourth was integration with existing industry tools. So the first part of my project was data acquisition. So I used a dataset called the DREAM dataset, which has 5 in silico time series networks, 1 in silico real-time network, and 1 real E. coli network with experimentally validated interactions. Gene expression is a good target because it's fairly easy and widely available across various different bacteria, making it a viable measurement from which to extrapolate GRN connections. So after I obtained the data, I preprocessed it by normalization, imputation, and feature selection, and I added small levels of Gaussian noise in order to reduce the biological redundancy associated with the data. The next part of my project was constructing a mutual information algorithm. So mutual information is a concept in information theory that essentially tells us, well, if we have, say, gene X and gene Y, how does having information about the expression of gene X change our knowledge about gene Y? And how can we use the information from that relationship to determine how much of a probability there is for an interaction between gene X and gene Y? So mutual information traditionally is extremely computationally expensive and is extremely infeasible because of that. So what I did was I modified the mutual information algorithm to be compatible with parallel processing. So I pursued a data partition with efficient candidate sorting and a KNN-based algorithm based on Kraskhov's methodology. And I calculated the Chebyshev distance in order to find the k nearest neighbor. So after that, I used the distance from the nearest neighbor's computation to construct a hyperrectangle and counted the points in the joint and marginal space for each gene in parallel. And I used the Kraskov's mutual information formula with digamma estimation in a parallelized inner and outer loop to estimate the mutual information for each gene pair efficiently. And at the end of this process, I attained a gene by gene aggregation metric of mutual information, meaning that I was just able to sum up the mutual information for each node to get a sort of measure of connectivity for each gene in the network, which could then be fed as an input to the GNN. So the third major part of my project was the construction of the interaction prediction graph neural network, and I used a spectral convolution-based, or more specifically, Chebyshev convolution-based network. Spectral convolution, unlike traditional Euclidean convolution, does not operate in the Euclidean domain. It operates in the spectral domain, and that allows it to avoid a lot of the intense eigenvalue decomposition and eigenmatrix calculation that is associated with traditional convolution, making it more efficient. And it also enables it to learn both local and global regulatory structures, which makes it more accurate. So the node features I used were gene expression and aggregated mutual information, with an edge feature of mutual information between each individual gene pair. I used my NVIDIA RTX 4090 for development, and I used an encoder-decoder framework with a dot product decoder and sigmoid output for interaction probability. I used a 50-20-30 train validation testing split, and the more detailed picture of the architecture can be seen on the right-hand side. So after I made the algorithm and the model, I realized that I had a lot of tuning and optimizing that I had to do before it could be used in the real world. So I performed several experiments on both the algorithm and classification task, and I optimized the Gaussian noise parameter and determined that negative sampling results in better AUROC and AUPRC score to balance out the imbalance in the classes between positive and negative interactions. I also optimized the learning rate and pursued cross-validation to ensure the model was not memorizing labels. The fourth part of my project was visualization and integration. So I made sure that Gemini was compatible with existing industry tools like Cytoscape, which it was, and I integrated it into a comprehensive backend framework for commercial use. So the first major result that I had was just the results from the mutual information algorithm. And we can see specifically from these graphs I pulled from Gao et al. And Zhao et al. That the KNN-based method converges to a low bias in the log-log plots and a low variance as well, which just indicate that there's low deviation from the true mutual information, which essentially is an indicator of quote-unquote accuracy. And the variance indicates that regardless of the marginal distances, the mutual information values are not too spread out from one another. We can also see the mean squared error here, and we can see the KNN-based method has a much lower mean squared error than von Mises and resubstitution, and the mean squared error actually decreases with sample size, indicating that the gene expression data is a good fit because it often contains thousands upon thousands of samples. The next result was a runtime analysis because I wanted to evaluate how efficient Gemini was. And I found that compared to Parmesan Arachne, which is the current gold standard mutual information inference algorithm, Gemini was about twice as fast. And compared to Genie 3, which is the current industry standard full GRN inference algorithm, Gemini's processing rate was about 9.5 times faster than Genie 3's on a 64 core parallelized GPU. That would take that human genome inference task of 2 years that I was talking about at the beginning down to just 3 months. And this indicates that Gemini outperforms a variety of mutual information algorithms and is significantly faster than what currently exists at the industry standard. So next, I wanted to evaluate the accuracy of Gemini. So I performed several benchmarks on the Dream4 dataset, which was the in silico time series dataset, and I evaluated Gemini's AUROC and AUPRC scores. So we can see on these 5 graphs on the left, with the x-axis being state-of-the-art inference algorithms and the y-axis being score, with the blue bar being AUROC and the red bar being AUPRC, that the AUPRC score was significantly higher than almost every existing algorithm. That just indicates that Gemini is correctly predicting the positive class. A lot of current algorithms are inaccurate because they simply overpredict zeros and are able to get a high AUROC or accuracy score because the majority of interactions are null, but they still result in inaccurate predictions because they disregard positive interactions. So Gemini's high AUPRC score ensures that it does not do this. We can see AUPRC score increases of about 300 to 400%, indicating that Gemini is outperforming all other existing algorithms with a comparable AUROC score as well. So next, I wanted to see if this effect was different on time series or solid state data, and I found that we observed very similar trends on solid state data. Gemini is one of the few algorithms that can perform on both and performs well on both as well, indicating that it's not constrained by data type. So next, I wanted to evaluate how applicable Gemini was in the real world. So I evaluated it I evaluated its performance on E. coli, which is the most commonly used bacteria in the genetic engineering space. Its AUROC and AUPRC scores on the experimentally validated data were extremely high at 0.95 and 0.93, with additional validation from the MCC, Cohen's kappa, Jacquard index, etc. I found that the recovery rate of known interactions was about 98%, indicating that Gemini performs extremely well on biologically plausible networks and is thus applicable in the real world. So, Gemini was also able to predict about 468 completely novel interactions. And I cross referenced all of these interactions with literature and found that the top 10 interactions were highly correlated with sources from literature that indicate that these interactions might be possible or even likely in E. coli, indicating that Gemini not only performs well on experimentally validated data, but is also able to outperform existing experimental validation techniques by predicting completely novel interactions. So I was able to use those novel interactions to construct the most complete gene regulatory network of E. coli to date for widespread use in industrial scale genetic engineering. In order to develop Gemini, I followed the product development, economic feasibility, and validation with advanced software high level development flow. And I was able to talk about licensing Gemini to local biotech firms and receive validation from Vanderbilt, Auburn, and UCSB. So to conclude, Gemini had a significant breakthrough in performance with a very significant efficiency improvement in AUROC and AUPRC score improvement. It's accessible, scalable, and easy to use as it's fully end-to-end and is open source. It's also low cost because it removes reliance on supercomputers and is industrially applicable as it performs well in real data. It also has several cross-disciplinary applications because the mutual information algorithm and GNN are applicable to any field involving causal inference discovery, so all design goals were met. So in the future, I'd like to expand Gemini and address its limitations, which were mainly that Gemini only relies on gene expression data, so may miss high-level connections, and that predictions are reliant on computation, and so they need further experimental validation. I hope to integrate other types of data and pursue wet lab validation, as well as industrial testing with small biotech firms, which have already been identified, and integrate it with AI services for contextual biomanufacturing and other highly industrial settings. I'd like to thank all of my amazing mentors at the Academies of Loudoun for helping me with my project, and that is Gemini.